We thought that given all that's going on, particularly with immunotherapy here at Penn, that it made sense to do something a little different and shine a lantern for you all on all the different things that are going on within Penn. You are in store for quite a treat this morning because we've been able to assemble, we're very thankful for all those that came, quite an impressive group of people that you're going to hear from this morning. We're very thankful to be here at the Penn Center for Innovation for Pennovation. I'll talk a little bit more later about Pennovation and PCI. What I did want to recognize is that this is not a fundraiser. However, if you are moved from what you hear, we're very thankful to the Cancer Center and the Cancer Center Development's Office in helping to arrange a number of these speakers. And I'll tell you why it's important. A lot of what as you'll see here today, and the people that you've seen here today, have been funded, not just by government. Yes, government's a big funder, but by individuals. And one of those individuals is Richard Vague, who unfortunately couldn't be here today. And you probably know Richard is an entrepreneur extraordinaire, Penn trustee, an individual who's actually recently won the Pact Enterprise Legend Award just last year. But Richard is a phenomenal individual and has been a huge supporter of what's been going on within immunotherapy at Penn. And we're very thankful to have the leader of the Cancer Center's Development Office, Tricia Bruning, who actually started the Cancer Center's Development Office and really has been a factor in helping to advance this technology with the capital that she's been able to raise. So please, a round of applause for them. And so with that as a kickoff, I'm going to get back up and introduce our first two speakers, but we want to show you a video first to really set the stage.
When you think about a revolution, you think nothing will ever be the same again. And we feel that way with immunotherapy for cancer.
April 11th in 2012, and that's the day we treated Emily Whitehead, our first pediatric patient with CAR T cells.
She had a very high fever one night, and when it broke, she had no evidence of leukemia. Her T cells were working.
Now we have the hindsight, almost 5 years later, she's still in remission and she's a normal child.
What's exciting for me is to meet patients who've been treated and to see them go back to their everyday lives.
Every single patient I have seen go through this has gone through this with courage and dignity and really have led the way and provide hope to so many more people.
It's changed the way I practice surgery in the way that I'm much more hopeful, so it's really exciting.
So the therapy that we started here in Philadelphia was initially 3 patients from our local Delaware Valley catchment area. And now our trials are international. That's really an exciting event and just the beginning of globalization of this new kind of therapy.
We have seen a remarkable thing happen in cancer immunotherapy here at Penn. That 20 or so years of basic immunology discovery led to the tools that could make this kind of therapy today. So we can now build on these cancer discoveries and these cancer successes to use the immune system for many other diseases as well.
So we're all interested in waking up the immune system and using different elements of that immune system to develop therapies.
We have everything here. Really, I think it's the only place right now in the United States where everything from conception of the idea to delivery in humans can be done in just one institution.
There is a unique spirit and unique group of people here at Penn that really has allowed this to happen here, to happen now. The idea that this type of therapy can be developed in the course of 7 years from the first patient treated to FDA approval is astonishing.
Quite honestly, the FDA approval of CAR T cells means that yes, we can win this. We can do something that's never been done before.
The FDA approval really gives a mark that we've made something happen, and it happened here in Philadelphia at the university, which is an incredible landmark.
It's like landing on the moon, pretty much. You know it could happen, but you never thought it could happen. So there we are.
All the components are in place to be able to make these great breakthroughs. This goes way beyond cancer. What we need, frankly, are resources to do this faster and more efficiently.
Philanthropy has a role now to accelerate this so that it becomes universally available as rapidly as possible.
So here's our promise to you. We're going to move fast, we're going to be impactful, we're going to get this done, and we're going to improve the lives of patients.
For a conversation much deeper about everything you just saw, please help me in welcoming our two speakers. We have a Penn trustee. Osagie has been an incredible entrepreneur in this region, giving birth to many very successful companies. He's got his start way back in the day at DuPont. He helped to start GSK Ventures. We're very fortunate to have him here today interviewing Dr. Carl June, the gentleman who's responsible for much of what you just saw. So please join me in welcoming Dr. Carl June and Osagie Osagie.
Good morning, everyone. So, I'm just the sideshow. Here's the star. Carl, I'm going to ask you a number of questions. Some of it have been questions that people had and sent to me. Some of it are some of the things that we've been thinking about ourselves. So let's start off with the timing of this. It's fantastic. I read a front page article in the Enquirer this morning about some of the work that you and your colleagues have been doing. So how did you get around doing this? Could you tell us a little bit about this CAR T?
You know, it's a 30-year story. So, but the long and short was the last disruptive therapy for leukemia, which is what I trained in Seattle, the Fred Hutchinson Cancer Center, was a bone marrow transplant. And that never got FDA approved, but it was something they said was impossible to globalize because it was the first personal cell therapy. You had to find someone to be a bone marrow donor. And then they got horrendous chemotherapy and radiation. And when it worked, it could cure leukemia. But and it was only done in two places, and it was Seattle and Johns Hopkins, where I was yesterday. And you know, it was said it could not ever be made generalized, and it happened. But it took 30 years, really, of training people, and then that it eventually went to community hospitals. So then, while I was there, I saw the power of that, which was a new immune system, and then it could, if it didn't kill you with what was called graft versus host disease, it could eradicate the leukemia. So what we found out how to do over the 30 years was to make your own T cells do that, which is the cells coming in from a bone marrow donor. We learned how to engineer them, and then they're your own cells and not the donor, and then there's not graft versus host disease.
So you said it took 30 years to get here, but how come this happened at Penn? What is particularly unique about Penn that has allowed this to come together at this time?
Yeah, so Penn it's not an accident. So I was in the Navy for 20 years, and then when I left, I got offers to move to Harvard and to Penn and to many other institutions. And I decided, Penn had made a commitment in the early 1990s strategically to make cell and gene therapy a primary objective. They recruited Jim Wilson, and then he recruited me in 1999, so he came about 1994, 95. And so there was a big commitment. It made the infrastructure, and we filled in the holes that other institutions didn't because of that focus of a translational endpoint from basic discoveries.
I obviously am quite enamored of Penn. I joke that I bleed red and blue. But we are part of a bigger infrastructure. So you've talked about the uniqueness of Penn, but how does the Philadelphia community, why this in Philadelphia? There's now talk of Philadelphia becoming the Silicon of the East, which I kind of like. So why Philadelphia? How do we pull all this together?
Well, I had never really been here, but What Penn has is it's a giant matrix organization, so you can put people together relatively easy. It's not a stovepipe place where people tend to work in their own foxholes. So that was a major advantage. It came out of this Quaker institution, and that's how they worked. Another advantage was, you have to remember, in my generation growing up, I was the first group of, kids born who didn't have to die or get polio. And that was because of this, the vaccine that was made in Pennsylvania. And so it had a history there. You had to grow eggs to make the vaccines and made the polio. And we got GSK, for instance, made vaccines that were not ever made before, and that required a certain infrastructure to do that. So that was established here in the Delaware Valley, and that's led into, it really synergizes with cell and gene therapy efforts that we have now in this emerging Silicon Valley. So there's a very long history of 50 years probably that led that. That didn't happen in the areas of the West Coast where it was IT.
So let's take this to the third level. So you've got Penn, you've got Philadelphia and Pennsylvania. And then how are we handling this on the international level? How are we exporting knowledge? How are we importing knowledge that supports the kind of unique work that you're doing?
We had the first successes. A lot of that we had the infrastructure, and then we had some really critical philanthropy that allowed us, our initial therapies could not be supported off government grants, and there was no industry. And so people, like the Abramsons made a huge contribution. That's why I came here, was that donation that allowed the Abramson Cancer Center to really become what it is now. It was very different, back in 1999 before that transforming donation. And then now we have all these international patients coming, and it's created, both it brings in new patients and it accelerates our trials, and we have impact now all over the world. I had last month, one place where it hasn't touched yet is the southern hemisphere. And that's not the emerging market that the pharmaceutical industries go after. And I went down to Costa Rica and had dinner with the president of Costa Rica. He took a selfie with me, and they have 60 kids a year who get leukemia in Costa Rica, and they're not going to get therapy, but we can do that with the infrastructure we have here, and we're working out where we can ship cells from the National Hospital, Children's Hospital in San Juan, and to here we manufacture the cells and ship them back. And so that will, that brings out our technology. Later, as the economy changes, that'll happen in an industrial way, but it wouldn't happen any other way now without the reach we can have through this.
So we've talked about three ripples that you've been involved with, Penn, the larger community, the international community. I'm always fascinated. How did that first pebble start? How did that first pebble hit the water? What inspired you to go into science, and what inspired you to do the kind of work you do now?
I thought, I think you just have to say, I was always interested in science, but I was interested in a lot of different things. And then seeing how medicine could change someone and help them. It was a huge thing. So I saw that early on. And then it all just changed. You have this set of opportunities. And for me, my life, I thought you can't plan things. So I was initially going to Stanford because I grew up in the San Francisco area. And then in 1971, I get a draft number of 50. So I ended up spending 21 years in the Navy. I never thought, but there I learned all about HIV and how to vaccine therapies, from the Army and the Navy, so I learned a lot on that and spent 10 years studying HIV, and then we ended up using that for how we engineer the cells for cancer patients. So I never would have picked that path. It turned out it was ideal to give me the unique set of skills. I brought Bruce Levine from me when I came here in 1999. He had come to my lab in 1992 at the Office of Naval Research to do HIV research. And he's here now. He's a professor, and he runs our, the cell and vaccine manufacturing facility here. And so it all came initially because of HIV, and then now it's cancer. So you can't plan things like that, is one lesson. Another, I think, is perseverance. Everyone said it could not be done. Initially, they thought we were crazy just trying to make the immune system go against cancer. And then, so we couldn't get funding. Fortunately, philanthropy let us start our trials. And then it worked. And it worked in an amazing way, and it was clear it could work, but everyone said, it can never be a commercial product. There was no industry to hand it off. And so there are a lot of people here that helped that to happen. And the tech transfer here, there was no industry for cell and gene therapy. It was amazing. And then now it's huge, and it's global.
Carl, this morning there was an article, front page of the Enquirer, about a publication that was just released yesterday in Nature Medicine, and I'm sure there are many more coming, about this unique subset of T cells, memory T cells, that could both act as a screening capability for people who would be more responsive, and could possibly become something that could be engineered within your shop. So what keeps you going? You guys keep on doing some fascinating stuff. What keeps you going?
So, that's an example where, in fact, some of the people in that movie we just saw were, they were the first leukemia patients we treated in 2010, and we studied, it's turned out it's a living drug, so now we have them 7 years later, and what we reported yesterday in that paper was that those cells are still working in them 7 years later. So it's a true living drug, and that's what sets it apart from all of the pharma industry. And what keeps us going now is the blood cancers, I think, it's becoming an engineering issue. I'm pretty sure we can cure every one of them eventually. The time is how long. What will completely disrupt oncology is that within a year, I think, myeloma will be FDA approved with CAR T cells. So myeloma is the most common blood cancer in adults. Our initial indication is pediatric leukemia, age 3 to 25. 600 cases a year in the US, that are refractory. So that's an ultra orphan. But myeloma is 2% of all cancer patients and 7% of all the financial costs that go in the United States for cancer therapy. So there's a lot of financial toxicity with myeloma, and it's not curable. Complete will change oncology, late next year when that gets FDA approved. Then the major issue is, solid cancers, the rest, lung cancer, breast cancer, and that's where we need help. That's a big issue. I am confident it can do it, and that, the therapy will work for all those cancers, but it's going to take a lot of work, to get that path worked out. And we have trials now in solid cancers like prostate cancer, pancreatic cancer, and so on, just to get our toes in the water on that.
Now that we've got this extraordinarily exciting momentum, what is missing? What can we do? What should we be doing? How do we get more Carl Junes? I know we can't clone you yet, working on that yet, but what else should we be doing as a community to make sure that we can create more Carl Junes?
So, that's a major effort that I have now, and I think Bob Vonderheide in our cancer center, and, is to retain our best trainees. So that's, right now there are, Penn has been the source of most of the innovation in cell and gene therapy. And so we want to keep the best and recruit in the best. That's been the key of what happened in Seattle at the Fred Hutchinson Cancer Center, and it's lasted 30 years. So that needs to be a major effort, so we need to grow. We have one example, I mentioned prostate cancer. So our partner Novartis is not really interested in that. It's too far down the road. But after our paper came out, the first 3 patients we treated in 2000, 10, it was reported in 2011 on August 11th. On August 12th, Michael Milken called me and said, how can we get you to work on prostate cancer? And so we started working on that, and then that's led now to we're getting a team to work on prostate cancer. George Weiss, one of our trustees, I said, we can't get funding on this from Novartis, we need help. And in 72 hours, he raised $2.5 million. And we have a prostate cancer team now, and we need to do that for every cancer. So we need to get the teams together, and then it will happen.
And what would you consider, I know we want to get on with the rest of the program, but what would you consider the biggest challenge that you're currently facing in your efforts?
It's hard. I think, it's a scale issue. I mean, unfortunately now we have with Tmunity, and you're going to hear from Azam about that. I mean, we have ways now where there is a tech transfer handoff. We didn't have that before. It was enormously frustrating. And now it's there. I mean, it's a $10 billion industry for sure, and growing in the cell therapy area. So we have that. So our challenge now is to stay focused on what the unique aspects we can do at Penn here, which is those initial tests and finding 3 patients like we did in leukemia, and doing that in brain cancer, doing that in pancreatic cancer, et cetera. And that's what we have to be focused on, because then it'll then go out now into tech transfer.
Carl, this has been fantastic. I cannot express strongly enough the time you've devoted to this dialogue. Thank you very much, and I'm sure the audience would also express their thanks to you. Thank you. Yes, good. I didn't know whether we could do that. Excellent. So, questions?
The question is about, the cells are hanging around for 5 to 7 years. What are the downsides, really? Transformation of the cells could, because right now, for instance, we have a trial with CRISPR. It's the only trial open in the United States, and it's here. And we're editing 3 genes at one time in a patient's T cells, and then also inserting a lentiviral vector, this HIV. So it's 4 genetic modifications at one time. So that could cause later the cells to transform, become a T-cell leukemia. So we watch, there's FDA regulations for 15 years, the patients. And the safety record so far is with over 1,000 patients treated, not a single patient's had that happen with our CAR cells now. And so we will be watching on every trial for that. I mean, so those are the 2 main risks. And it's safer than chemotherapy. If you look at women who have had chemotherapy for breast and ovarian cancer, about 5% get what's called secondary leukemia, AML, which is an awful disease. So it's much safer than chemotherapy, but we're still early days here and don't know all the risks yet.
Yes, thank you, Dr. June. You've had a lot of success with blood cancers, and what is the future for treating solid cancers, which I think are trickier?
For sure. I mean, the solid cancers are more complex genetically, and they have evolved more ways to shut down the immune system. What we know is, and I'm an optimist, so I always look at how things can work, and not necessarily. But what we know is if we take cancer cells out of a patient that have, from any solid cancer you can imagine, and we've done it in almost all now, and make a CAR T cell in vitro in a petri dish, the CAR T cells win. So they kill the cancer cell from basically any cancer there is. And so the issue then is in a patient getting enough cells there. And it's what we found in leukemia is it's basically a game of numbers, and that the reason previous therapies failed is they kill one by one. And so, a kilogram of cancer is 10 to the 12th cells, basically, if you look up in Wikipedia. And with the patients we treated all had at least a kilo, 2 pounds, 2.2 pounds of leukemia or lymphoma. And we would infuse 100 million T cells, and in the space, we know Emily Whitehead, in 14 days, she grew 10 to the 12th CAR cells in her body. So that's a kilo of T cells. She was a bioreactor. It's a huge metabolic stress to make that many cells. But that one-on-one, then they killed the leukemia. Then what they do is most of the CAR cells die, but some live, now we know, out for 7 years. We need to do that in solid cancer. We need to make the CAR T cells be able to divide when in the tumor microenvironment of a solid cancer is it's very low in oxygen, it's acidic, and it's toxic to the immune system. So we need to make them armored, the CAR T cells, so they resist that. And we have all kinds of strategies to do that. And I think the main issue is how long it will take. And if we can get more people working on these things, it'll happen more rapidly.
We have a question right here. Hi, thank you, Dr. June, for your work and for creating new paths and new hope for everybody with cancer. I'm Helen Loeb from the Children's Hospital of Philadelphia. My son was diagnosed with cancer of the esophagus. That was 2 months ago, and he's undergoing treatment at Abramson right now, chemotherapy. So I guess my question might be more on the kind of the human side, like with all I found myself like researching, cancer and the news and reading all of that. And so with all the hope that's coming and, people are reading about immunotherapy, with all the pressure, how do you manage all of that when there are so many people who ask you, when is it going to happen? When is it going to happen for solid tumor cancer? So how do you manage the pressure?
Well, that's it's a very difficult issue. I get emails every day from cases like yours. I'm sorry for the challenge you have with your son. On one hand, when we started this, the most common cause of death in kids from cancer was leukemia. And now we've changed that. Now the most common cause is brain cancer in kids. So the natural history of cancer in children has changed, and I think that gives us resolve, to work on these others. Rare cancers like esophageal and a child is very rare, and Children's Hospital is the best in the world at, doing research and to make that challenge be solved. What we have is the largest team of people doing this immuno-oncology now in the world here. But it's the big unknown is how long it will take, and the more people we can get on this, the faster it will be solved. We worked very closely with Joe Biden, who lost his son to brain cancer. And
thank you. So it seems like these therapies, there's a lot of different individual pieces in creating these cells. There's choosing the cell type, the effector cell type. There's the question about allogeneic versus autologous. There's a question about the particular kind of genome editing and then the particular receptor. Have you thought at all about what the ideal sort of target product profile of an off-the-shelf sort of mass-produced cell as a product is going to look like, say, 10 years from now?
Yeah, we're working on that, and as are others. So, I think what we're going to have is two kinds of therapies 10 years from now. One will be the patient-specific cells, which are going to be hard to design a better cell than the one your thymus did through Darwinian evolution, and it can live in your body your whole life. Okay, and we can genetically modify that, and that's safe. But not in every patient can you make those cells, and it takes time, and it's expensive in a cost of goods manner. So the off-the-shelf cells could come from a variety of sources, such as cord blood or other healthy donors that don't have cancer, and things like iPSCs and ES cells, et cetera. So there's a lot of basic advances in that right now. It's not a commercially viable approach. It's a very long, extensive culture, and then they haven't lasted or persisted in this patients for long term. And I don't think, in fact, you want to. There'll probably be a safety risk. So I think what we'll have is, as we have in other kinds of cancer therapy, induction therapy, and then maintenance therapies. And the maintenance therapy would be your own cells that are engineered, personally, and then off-the-shelf ones could be given in community hospital settings, and much simpler logistics. And so I am, those have already had effects in leukemia, but they're a bridge at this point to then another definitive therapy, such as a transplant. And we'd rather have that not have to happen. Could we have autologous cells then after the initial off-the-shelf cells? So I think they'll be very complementary, and then the off-the-shelf cells are much more in a standard pharma model. They can be made in large batches and much simpler logistics than what we're faced with, in making personal cells.
A very related question to that one. Your story about Costa Rica was fascinating. How do you see that evolving? Is it with this more of this maintenance kind of core blood type model for using it in a lot of centers right now? Because right now it's only used in eight centers, or potentially. So how is that going to expand?
So right now Novartis has more than 30 centers open where, leukemia patients and very shortly, lymphoma patients will be treated. So they're coming online after they're certified. People did not think it would be possible to globalize this therapy. The trial that Novartis did, was about 80, patients with leukemia, and they imported exactly what we've done here at the University of Pennsylvania, and cloned it into Morris Plains, New Jersey, and then conducted a trial globally, and they had exactly the same response rate we had, and they didn't have a single death from cytokine release syndrome. They had 48% incidence of that, just like we do, but they trained people very carefully how to do it. So that was an amazing result. No one thought that was possible. The issues in Costa Rica and, India, Africa, and South America are huge global health issues. This chain of supply and so on needs to be, it's very difficult logistics for there. So that's where philanthropy can really help accelerate that to happen and make the technology come out more rapidly available.
Can you please describe the moment or the time period where you realized that this was going to be successful and against all the odds, and you and your team?
Yeah, it came out over a period of 6 weeks in 2010. So we treated the first 3 patients, and all 3 responded, and all 3 were literally, on their deathbeds with, advanced leukemia, and had no options. And so, I didn't see the first patients for conflict of interest reasons. I worked on making the cells with our team, from the bench side, and David Porter and some of his colleagues took care of all the patients here in the Cancer Center. And the first patient, Bill Ludwig, who was on the front page of the New York Times, and many times he's been here, we didn't know what was happening. We treated him, and then within 4 days after treating him, he was in the ICU. He got to the point where he got last rites. We thought he would die. And we didn't know what cytokine release syndrome was. It had never been seen. So it was a hyperactivated synthetic immune system, and he had 104-degree fever for 2 weeks, and they thought it was some infection that they couldn't diagnose because there never was an infection resolved. And finally then, he got through kidney failure, lung intubation, all these things in the ICU. And when he woke up, they finally did an exam on him, because until then, they were just worried about keeping him alive. When they examined him, they couldn't find the tumor. And he'd had it growing out, you could feel him and explain there was pounds of tumor in his spleen. And so they did a bone marrow aspirate, which is where, the bone marrow of the leukemia starts, and it came back no leukemia. And this was 28 days after we treated him. I said to email, I said, David, this can't be right. Would you do it again? And so on day 31, they rebiopsied him on the other side, and it came back the same answer, no leukemia. So then we said, maybe this is real. And then we were dogged with this thing that lasted for about 2 years, which was, is this just some kind of fluke? The first patient, you always get this miraculous result. But the other 2 patients responded. Now we know, as of this paper that came out yesterday, 7 years later, they're still in remission. I think the patients regard themselves as cured. And we can't find leukemia in them. So we need longer to follow up. So it came like that. We did not expect, it worked actually better in humans than it did in mice. where and that is really, there's no precedent for that. And the mice didn't get cytokine release syndrome or any of the things we saw, the high fever and all that. But the leukemia did go away, but it wasn't as striking as what we saw in the humans, So, and then it turned out to be reproducible. We never thought it would work in children. Acute leukemia grows much more rapidly than chronic leukemia, which is what Bill Ludwig and the initial adults had. There, the average age in the U.S. is 70 when you get that, and then we treated Emily, and it worked even better. 90% response rate, and we had no idea that would happen.
The good news is that you have a powerful partner, Novartis, and I am assuming that's somewhat monogamous, so when they tell you they're not interested in this current inquiry, are you allowed to sleep around, or is it, could you find?
So we had, John Swirly worked on this. It was a very difficult issue back in 2012 when we signed this monogamous relationship with Novartis. And we did that, quite frankly, because, and we had many offers back then to spin out a biotech, but ethically we thought we needed to get this, how could we get this done most rapidly because of the pressure we were having, the patient demand. And Novartis had a major franchise in leukemia. That's what they made their name with, imatinib, Gleevec. So that's how that happened. Now back, fast forward now to 2018, monogamy is not the right way to be when you have a rapidly evolving technology in many different cancers, because there's no one company that can definitely, can handle that. So that's, we have Tmunity and there's biotech now has a very large role in this innovation now.
Hi, Dr. June. Thanks for coming, and thanks for your work. My question is regarding solid tumor. What about working with something like pheochromocytoma that's a single-driven mutated cancer? Would that be an easier step into the solid tumor world?
We're working on it. So, in fact, I have this awful story. One of the postdocs in my lab, his 32-year-old wife developed a neuroendocrine tumor last year. So. And it's a tumor that does not, it's a Steve Jobs-like tumor. It does not respond to chemotherapy. Right. And there's some complex reasons for that. And so we are working. I have relationship with. We have grants here now from the Neuroendocrine Society to work on those tumors. And so it's they're rare, but they're a large unmet medical need for each person who gets those. And again, pharma is not really interested in that. It's a long-term project. So we're working on it, and we think, I mentioned that, if we in vitro, we can kill those cells with a CAR T cell. So in a patient remains to be, we're working to get there in a clinical trial.
Thank you for being here. As therapeutics evolve for oncotherapy, I think about all of the successes and all of the failures that we've had. So immunotherapy that's out on the market today is great for those for whom it's great, but unfortunately the population for whom it's great is still very small. So I think about the other treatments. Can you give some insight perhaps on how you think that will evolve, particularly with the evolution of your therapies?
Yeah, so there has been a glass ceiling, if you will, with the checkpoint therapies. And depends on the tumor. Some don't respond at all, but in many cases it's 20%, leaving 80% don't benefit. But for the 20% who do, it's huge. And so how do we move that up? In acute leukemia with our CAR T cells, it's 90% initially get in remission, and I think that's what our goal should be. And so we do have trials now, both in Children's Hospital and our adult side here at the Abramson Cancer Center, combining checkpoint therapies with CAR T cells. And in our mouse studies, those work better than either one alone. So that's going to be one route. We also think that's going to be possible to do by just genetically engineering the cells so that they're checkpoint resistant. And that's part of our trial that we're doing with CRISPR. So, we have data, and others do, that combining some forms of chemotherapy or targeted therapies. So Sarah Gill in our cancer center has a trial with ibrutinib, which is a targeted therapy that targets the BTK kinase in leukemia cells. That is very synergistic in a number of trials we've looked at. Steve Schuster has a trial here in lymphoma, and we have it in CLL, and we're going to be pursuing that as a registration strategy with Novartis. So that's a Janssen & Janssen drug combined, targeted agent with a CAR therapy. There'll be CAR therapies with, antibody therapies and chemotherapies, radiation, checkpoint. So that's part of the scale issue we have is when you look at the possible combinations, and then, we need large numbers of trials to figure out how to do these. But we have striking results now in some of the trials where we've combined them. And so that's a very exciting possibility now that we have this one out in the market. Now we have a backbone we can test against to look for synergies.
Thank you, Dr. Carl June, for being here, and again, really enjoyed the talk. There's been a flood of checkpoint inhibitors in the market, and hopefully there's a flood of CAR Ts for different cancers. But where else do you see in the immunotherapy space, CAR NKs, protein-based, DNA-based, where else do you see innovation that might be relevant to what we're doing and hopefully bringing more therapies to the patients?
Yeah, so as you mentioned, there are all many kinds of cell therapies, and I think, and both for autologous cells and engineered cells. And, we will have one here, again, with Sargil and NCI and Tmunity, where we're going to have the first dual product using an engineered stem cell and an engineered T cell, and we're going to do that for acute myelogenous leukemia. So that's just one example. Oncolytic viruses, are a huge potential now. A number of companies are doing that. Jim Wilson has a lot of expertise in that area here at Penn, so engineered viruses like that have very much complementary activity with CAR T cells, for instance. So there'll be, it's, a huge exploding area. I looked, clinicaltrials.gov a month ago. There are over 270 different CAR T cell trials now in the world. The most are in China, actually. And then we're number 2 here in the US. And that's an issue on national prioritization where, we're not investing as much as they are in China, unfortunately. So, but there is this very large evolving global effort now. And, I think what we have here is a lead in those initial testing, and then there's global industrialization of this. It's such an amazing thing happening in a very short period of time.
Well, Paul, thank you so much for your time.
Not that you need evidence, Dr. June, but a raise of hands of anybody in this room that has been affected or knows somebody affected by cancer. I think it's pretty much 100%. Dr. June, we are grateful for your life's work and so indebted for you to be here today to share your story, and Osagi for your presence as well. So please, another round of applause. One of the most astounding things I just heard, as Dr. June knows, there are many scientists that have cured cancer in mice. And to hear that this therapy works better in humans than mice is unbelievable. It does take a village, beyond the walls of Penn. And here at Penn, PCI, the Penn Center for Innovation, has been a critical partner in the commercialization, not just of this technology, but many technologies. And we're very fortunate to be here at Pennovation, but also to have join us today many of the people who make that happen on a daily basis, including Mike Dishowitz, Mike Poisel, Dr. Mark Turco, Lori Ackman, sitting in the front row right here. And I'd like to now introduce John Swirly, the fearless leader of PCI, who is going to kick off our panel discussion with a couple of very interesting researchers as well as commercializing CEOs. So please join me in welcoming the head of PCI, Dr. John Swirly.
Good morning, everyone. What a great turnout. And thank you, Carl, and thank you, Osagi. that was very incredibly inspirational. My organization and I have been privileged to work with Carl and his team, since those first results back in 2010. In fact, you ruined my summer vacation that year, so thank you again for that. But it really is quite an amazing story and an amazing journey, and frankly a very difficult act to follow. So thanks for that as well. But we're going to give it our best shot here, and we have a phenomenal industry that we now talk about as sort of a given that this field of CAR and cell therapy it didn't exist. There wasn't a vibrant biotech industry in this space. The big pharmas were not yet committed to developing programs. It was extraordinarily difficult for Carl and his team to raise basic research funding and clinical support to do these incredibly important studies. So I say that because the impact of Carl and his team's work on the patients is evident and is absolutely tantamount. But the impact that this work has had on the development of the industry is equally impressive. And so I use that as an entree to the panel discussion today, because I think what you're going to hear from the group today are ideas and initiatives that have been enabled by this work and are going to further extend by logarithmic parameters the full potential of this space. So without further ado, I do want to introduce our panel. We have Dr. Amy Payne, the Albert Kligman Professor of Dermatology at the University of Pennsylvania, Dr. Osman Azam, who is the CEO and president of Tmunity, and Dr. Stephen Kelly, who is the president and CEO of Carma Therapeutics. So I'll invite these three up to the dais, and we'll get started. So I'm going to try to balance this out a little bit this morning. I'm going to give each of the panelists an opportunity to introduce themselves and their work and their objectives in their respective roles. I have a few canned questions that I'll walk through, and then I'd like to try to leave some time for questions. So if somebody can just give me the high sign when we're getting close, I don't want to cut out the important questions from the audience. But why don't we start with take a couple of minutes to introduce yourselves.
Hi, I'm Steve Kelly, President and CEO of Carma Therapeutics. My background is about 30 years in biotech. The first half of my career was in commercial roles for established companies, worked in supportive care, worked for Amgen. I was head of marketing for IDEC Pharmaceuticals that launched Rituximab and for lymphoma. I worked for Sanofi Syntalabo and launched a drug for colorectal cancer called aloxatin. And then around 15 years ago, decided to switch and become more entrepreneurial, and I've done a series of startups, about 5 different startups. The last 8 years or so, I've been in the Boston area. And on February 4th, we were watching the Super Bowl, and at the end, I turned to my wife and said, this dynasty's over. Let's move to a city of champions. and so a week later, I accepted the job at Carma. No, seriously, Carma is a very exciting company. We're working, I would describe it as the intersection of adoptive cell therapy, which I think is, as you looked at cancer therapy, moving from small molecules to antibodies, and now adoptive cell therapy is really the next renaissance, I think, in cancer treatment. The CAR technology developed here, as well as macrophage biology. And so what we've done at Carma is taken a CAR and attach it to a macrophage. And a macrophage is obviously different than a T cell, has a different mechanism of action, and we believe is ideally suited for the treatment of solid tumors. Macrophages traffic to the site of the tumor and use a different mechanism of action, phagocytosis. Essentially, it's the body's Pac-Man, and it goes through and eats up different cells. And so we have targeted against a surface antigen on a cancer cell and directed it, and we believe that will be quite beneficial to solid tumor patients. We're early stage, probably a year and a half away from the clinic, but we're super excited about it, and I'm super excited to join the Philadelphia biotech community.
Good morning, everybody. My name's Oz Azam. I have the privilege of being the president and CEO of Tmunity. Tmunity is a T-cell engineering development company that's focusing on the next wave of technologies, be they CARs or TCRs, focusing primarily on oncology, but we're also looking at conditions such as HIV and hopefully also autoimmune disease. By way of background, I've been involved in innovation and life sciences for about 20 years. For the past 10 years before I joined Tmunity, I had the privilege of holding R&D roles and commercial roles at Novartis. And back in 2013, I had the distinct privilege to get to know Professor Carl June, John Swirly, and many other colleagues at Penn, and was involved with the delivery of the first generation of the CAR-T programs, namely Kymriah, that Carl mentioned, that was approved for pediatric cancer. So I'm delighted to be here, and I look forward to John's challenging questions this morning.
My name is Amy Payne. I'm a dermatologist, and I specialize in autoimmune disease treatment, specifically in autoimmune blistering disease known as pemphigus. And my research area of interest is in autoimmunity. So I set up my laboratory a little over 10 years ago here at Penn to study the B-cell repertoire in pemphigus, because it's an antibody-mediated disease, and B cells make antibodies that cause that disease. And so many of the treatments that we use for pemphigus are very similar to what's used to treat B-cell leukemia. And so one of the mainstays of our treatment right now is Rituximab. So we pay a lot of attention to the B-cell leukemia literature. And when we started reading that Carl was curing B-cell leukemias right around 2011 and 12 when it was reported, we were thinking, how can we look at that and make that work for our patients as well? So what we did was re-engineer that technology. So whereas the CAR T-cell uses an antibody to kill an antigen expressing cancer cell, in this case CD19, we're actually trying to kill an antibody expressing B cells. So in that case, we can use the antigen that we know is targeted in disease, which in this case is something called desmoglein. So we actually threw the antigen onto the CAR T cell and used that to hone out and seek and kill only the antigen-specific B cells. So it's a way to achieve antigen-specific targeted B-cell depletion. It's really kind of the holy grail for autoimmune disease therapy, because our patients face many of the same problems that the cancer patients faced going in, which is that we used to kill just all of the cancer cells with rapidly proliferating anti-proliferative agents. But that caused a tremendous amount of toxicity, and that's the same problem our patients face now. So we basically have no way of targeting just the autoimmune cells, so we wipe out the whole immune system with things like Rituximab, and it leads to risk of fatal infection and other problems. So we're hoping to use that antigen-targeted approach to treat autoimmunity now. And I guess by way of introduction, I actually lived in San Francisco, St. Louis, and Philadelphia, and they all won Super Bowls shortly after we arrived. So that seems to be a good trend. Of course, they also had an earthquake and major floods, so I think I'm high risk, high reward.
Super. Thank you. Thank you, all three of you. So I guess I'll get us kicked off, and I think you're actually. The panel is maybe the answer to this question, but I'll go ahead and ask it anyways. I either saw a headline recently or someone asked me this question, and I thought I had to think about it, but not too long. And the question was something like this: it was, is the cell and gene therapy sector overheated at this point? And I think part of the reason that question arose is, that there, as again, kind of looking over the last 10 years, it's hard to pick up a journal or read even a mainstream medical science article these days without seeing something related to immunotherapy, cancer immunotherapy, cell therapy. And so I guess it's a natural question, but I'm interested in the panelists' reaction to the notion that cell and gene therapy is overheated as an area for investment, but also as an area for further research.
So I think that actually it's not. So one of the concerns is that as we get into larger use in the clinic, we see that there are problems. But as a researcher, that's the opportunity to better understand. And so if we can basically understand and actually have these patient cohorts where there are responders and non-responders, classic example would be Carl June's recent paper in Nature Medicine. They actually use that to better understand what exactly is happening, what are the natural predictors, and by doing that, you can actually figure out how to design the next generation of therapies and what we can actually target. So I think, if anything, it gives us more knowledge that we can use to basically better tackle these things, and obviously also target different targets with autoimmunity and solid tumors.
So it depends who you talk to about overheating, right? If you look at it from a market perspective or an investor perspective, then there's probably never been a better time than now. And it's driven by the science, right? And the proof of principles. So I think there's a school of thought, absolutely, whereby some would say that maybe there has been some overheating in the markets driven by companies that maybe prematurely IPO'd on the backs of platforms and technologies that weren't fully validated. Case point being some of the CRISPR technology companies that probably are now right-sizing as the practical applications of those tools are now coming of age. But if you look at it from a interest perspective of companies like ours, then we as CEOs probably would say, no, it's not overheated, because right now the science is flourishing, it's coming at a fast pace. And I think the difference is that investors and smart investors are now starting to realize, and I think Carl mentioned it earlier, right? The next wave of challenges for these therapies is really making them globally available and scalable. And for that, you need investment, and you need to invest early on rather than later downstream. And in our worlds, a big part of our lives are around manufacturing science enablement to make those products real. And you just can't do this as an afterthought when you've done something in an in vivo or in vitro setting at Penn, to then have an afterthought and say, well, hang on a second, how am I going to make this successful for a child or an adult living in Costa Rica, Japan, or wherever? You have to think that through earlier on. So the capital requirements for companies like ours have increased. And I think visionary investors see that, and they're prepared to come along for the journey in a company and build out companies. So it depends how you look at it. From my point of view, I don't think it's overheated just because of the challenge we've had ahead, but the proof of principle is there now, right, which is going to lead to further innovation and hopefully further investment.
I would agree. One way to look at it is when people talk about oncology or cancer, they think about chemotherapy. And chemotherapy isn't one drug, but a lot of people think collectively, well, that's I'm going to be under chemotherapy. I think that cell and gene therapy is a similar situation where you can't really bucket into one thing. You're talking about 300 different diseases within the oncology realm, and then different approaches as you go along. And so you have autologous CAR T, allogeneic CAR T, autologous TCR, allogeneic TCR. You have the different immune cells like we're looking at with macrophages. You have natural killer cells. There's a lot of different approaches and multiple and next generation approaches, like what Tmunity is doing. It's next generation to what has already been covered so far. So I think this is just the beginning. It's not overheated at all.
The momentum that's developed in this space happens because of investment and corporate partnering and participation in the process of taking these things forward. I'm intrigued, and this is a good panel to ask this question, because it's easy to see the large dollars flowing into companies and programs that are taking products to market. And I think all of you represent examples of instances where early stage technology, which is developed in the academic sector, needs to move into the private sector in order. And perhaps Amy, you can talk to us a little bit about your work and how you imagine that it is going to translate forward, and whether it is going to require substantial partnering and investment to do so. And I'm interested in exploring the relationship between the technology developers, the technology creators and the technology developers, and how that happens.
Yeah, so I think that one of the things that's unusual about cell therapy is right now it's expensive. The cost of goods are high, and for example, to do one vector manufacturing run, it's $500,000. Whether that treats one patient or 100, it's still $500,000. So it raises some issues in terms of figuring out how to best translate this into clinical trials. I do feel strongly that we need to take our data in mice and take that forward into the first human patients. The reason why the CAR T field exploded in a good way was because of those first 3 patients that were treated. So I think we need to do the same thing in pemphigus, basically, and take it forward to clinical trials, and that will really enable the entire field. So in terms of thinking about how to do that, that's a tens of millions of dollar investment if you really want to do it safely and properly. And so the question is can we still follow the same route of developing that through the academic laboratory with NIH grants and I think that that's a little bit challenging just because it will limit you in terms of how that works. So I think that partnership with industry is really the best path forward, where it can actually be developed and where you can actually be a little bit more forward-looking in terms of whether or not you're doing the right thing and the safe thing with patients.
John, I'll give you an example of I think what helps address your question. So I remember back in 2014, when I was at Novartis, we embedded a team of colleagues from Morris Plains, which was our manufacturing facility, with Bruce Levine and Carl. We parachuted a group of something like 30, 40 colleagues that literally learnt the process at Penn for the first generation of the CAR T that Penn had licensed to Novartis. And that journey took us well over a year to show comparability of the process that was happening in the lab to treat patients, to then get that scaled up in a GMP manufacturing scalable environment. And through that, I learned a ton, as you can imagine, right, of how to do these things well and how things can go wrong. But I think that the most important thing is that you know what you're good at and you know what your partners are really good at. And if you think about the ecosystem of companies like Tmunity or Carma and Tycho, all of us sitting here now, we rely on a model whereby we know what Penn is really, really good at, which is these nimble, fast proof of concept trials that treat many different patient types, basket trials, different types of cancers. But also Penn realized that in order to scale these therapies up, it takes a different mindset. It takes an engineering mindset, an industrial mindset, life sciences mindset to think through, how do you scale and create full value for a product and a platform, both from a commercial perspective and a patient perspective. So I think having an understanding and respecting each other's boundaries and knowing what your partner's really good at and where their gaps are and how you fill in those gaps. I mean, that was the biggest learning for me for an area that didn't really exist. I mean, until, Kymriah and with Kite, with Yescarta and Juno at the time, nobody really knew what they were doing, right? So it was uncharted territory, right? But we learned together, and through that blueprint now, we can advance things further and much more significantly in an accelerated way, hopefully. And I think also in addition, we've now got a regulatory framework that's really, really favorable for this sector. It's never been so favorable from an FDA perspective, right? Especially on the manufacturing side. So these are things that companies like ours really want to laser-like focus in terms of how do we, to Amy's point, really radically reduce the cost of goods of these therapies, because they are expensive, and they're expensive to manufacture. That's just because the raw materials cost a lot, and also treating these patients is difficult and timely. But over time, as you scale up and you treat more patients, and as the model becomes more efficient, then hopefully you'll realize savings over time.
And if I could add a little bit for Carma specifically, our 2 founders are at Penn, Sar Gill and Mike Lachinsky, and they had the idea to form the company, and they leveraged resources at Pennovation to do that, and working with PCI Ventures to form a company, and they got, I don't describe it as hand-holding, but guidance along the way on how to do it, in addition to small infusion of capital. The company reached a point where they needed to seek outside capital, which we're in the process of doing right now. But at the same time, we still draw on resources at Penn, and both the cell manufacturing facility, and we will look at them for clinical trials. So I think the supportive network within Penn has really helped the company form and grow and mature.
So I've been privileged to follow almost every one of your stories from the very beginning through to where you are now. And I can state unequivocally that every single one of these programs is founded around very elegant and top-tier science. I mean, these are top-tier publications, each and every one of them. They have a base of very intricate, very sophisticated science. And I think and this may be more, for you, Oz, and you, Steve, in terms of how you translate that breadth of knowledge and that complexity into a story that actually makes sense to investors, but without losing the thread, without twisting it in a way that it turns into something that it's not. If that makes sense.
So I'll take a stab at your question. So I think that the not losing the thread, right? I think the science is so rich, and it's moving at such a fast pace. It's really, really hard to keep track of it sometimes, right? So I think companies like ours, you have to learn as an entrepreneur, and trying to attract capital into a company. You've got to keep your story tight, and you've got to keep it consistent in terms of what we're trying to do, and stay very, very focused, right? I mean, the stuff that Carl described earlier on in terms of a 30-year journey, I mean, it now appears science fiction-esque, right? When you think about how do you take a cell and how do you reprogram it and how do you deliver, this amazing new molecule on the surface of a cell. But keeping that story tight and consistent is very, very important, right, for investors and for folks who are not investing, just the lay public, in really understanding both the strengths of your platforms and the limitations. I mean, we are literally on the cusp of a big inflection point in personalized therapies, but there's a whole lot we don't know. So I think also balancing it, and Carl very eloquently put it out there, there's a lot we know, there's a lot we don't know. And one of the fears certainly I have, and I'm sure my colleagues share that, is in this field, you can take 2 steps forward, 1 step back if you're not careful, right? Not by necessarily because of what you do, but because how the field and the science evolves. And science can be really helpful, and sometimes science can be your worst enemy as well, right? From a company standpoint. So agility of keeping ahead of where things are going, but also staying very, very focused and delivering what you know is your core value proposition and what you're frankly good at. I mean, companies like ours, we know what we're good at. We're good at T-cell development. We're really, really good at thinking through how to build commercial businesses around this and do these trials to scale. We're not going to be a company that's going to be a T-cell discovery company, for example. That would be a complete waste of our time. So we'd partner with Penn, we'd partner with others that are really, really good. So it goes back to really appreciating what you know you're good at and where you need the help. And I hope that gave you some perspective to your question.
Absolutely.
Yeah, and I'll add to that. As we've gone out and talked to venture capital investors, what are they investing in? They're investing in a big idea, they're investing in great science, and they're investing in teams. And so when we present our company, this is the big idea. We're going to commercialize CAR macrophages, and list out the ways that we're going to execute on a plan. And we're going to build a great team. Our founders are tremendous, and then we're starting to populate the rest of the team as we grow. And I think that as we structure how we're going to use the proceeds, part of it is to value inflection points so the investors can see a return. They want to see you're getting to this milestone, and that is a meaningful endpoint. But half of the money that we're taking in is building out our platform. So places that we can take our the initial technology and apply to different targets within oncology, and even potentially outside of oncology.
That's really helpful. And Amy, let me flip it for you. And I'm sure you've talked to potential commercial sponsors, partners about your work. Any lessons that you've learned in terms of maybe through trial and error, how to communicate in the powerful science in ways that is compelling and relatable to folks who maybe are not quite as immersed in it as you are?
Yeah, so it's interesting. I mean, in some ways, it doesn't necessarily change because as scientists, we're constantly trying to sell our science to grants review committees and things like that, and we always have to make it accessible at essentially a layperson level because, just because I understand pemphigus doesn't mean that somebody who is reviewing it at NIAMS who might specialize in osteoporosis, would appreciate pemphigus. So that you're always trying to reach at that level. And I feel that in some ways, you don't really have to change the story. My job as a PI and a scientist is to scan the project, see what my biggest fears are, rather than ignoring them, tackle them head on and just basically explore it and report what we find, So I think that ultimately, it really is aligned in the sense that that's the recipe for success. You know what your problems are, you address them head on, you're very frank about it. And then you also have to be your best champion. You have to be able to also say, but here are our strengths and what we're very excited about and what we think we can push forward. So I think in that way, the message is not very different from what Oz just mentioned. Know what you're good at, know what your weaknesses are, and find people that can help you with those problems.
That's terrific. I think we're running kind of short on time, and I don't want to leave the audience out. I think I've left it probably 5 minutes or so for some audience questions. Yep, and here we go.
Hi there. This is very interesting. There are a lot of incredible advances that are coming out that actually are revolutionizing healthcare. But the elephant in the room is affordability. And the pricing is understandable considering the risk to bring these to market. But what can be done to make it more affordable? Changes in funding, improvements in supply chain, differences in reimbursement. What can make this a more sustainable and scalable model for healthcare?
All of the above. I think, you know, and I'm going to restrict this to the world I revolve in, which is adoptive cell therapies and T cells. So I think in our space, it really is manufacturing and cost. It's a big driver of the cost and the burden, and that follows through to patients and healthcare system providers, right? But I do believe that there are, certainly within the next 5 years, you'll see some monumental progress on this, particularly in terms of the raw materials that are used, in terms of actually creating these products, the engineering that goes into developing and scaling these products, the enrichment of cells, the stimulation of cells. All of these things are now at a cusp and a tipping point where there are some great technologies out there that companies like ours are looking at and partnering with others in terms of actually really bringing down those cost of goods. And I think there's then a wider societal issue as well, right, in terms of priorities and where you focus healthcare spend, right? I was having this discussion with some colleagues over dinner the other night, funny enough, but I mean, there's a disproportionate situation, right, in society when you think about the market caps of social media empires, right, versus healthcare companies. I mean, as the pariahs of, you know, the healthcare industry, we're always slated, right, in terms of putting profit before caring about humanity. But I think there is a need to regroup and think about what our priorities as a nation, right? And where do you want to put your spend in research as well? So Carl said it earlier on, and he's absolutely right. The facts speak for themselves. Other parts of the world now dominate in terms of cancer research versus the United States. I'm originally from the UK. One of the reasons I came to the US 20 odd years ago was because I saw innovation and a spirit that I didn't see anywhere else on the planet with regarding entrepreneurship and regarding science, right? And now looking back 20 years, I'm now looking at, you know, where the world is moving to. And from a core research investment perspective, it's not the US right now. If you look at basic science and research, I mean, China is eclipsing us. That's something I think as a society we need to think about, right? In terms of where should we be writing to our congressmen and our politicians around where are the priorities in terms of research and development, right? So I think there's a burden on companies like ours to think through and do what we can. But I would push it back to society as well in terms of what do you see as your priorities, knowing aging populations, cancer's on the rise. There are new forms of pediatric cancers evolving all the time, new mutations occurring all the time. How do we want to look at this holistically as a society as well?
Oh, Lori has a question.
Thank you. Thanks for being here with us. So one of the reasons we wanted to work with PAC to bring this program together during Philly Tech Week was to expose kind of a newer and perhaps younger audience to all this innovation that Penn is doing in this space. Dr. June mentioned if we're going to continue to remain competitive, we need the scientific talent to grow here. And one of the promises the Pennovation Center is we're creating a landing pad for startups so we can really hold on to the Silicon Valley. You mentioned that you both were growing companies and looking for talent, and I just thought you could talk a little bit about the type of talent you're looking for and future opportunities you see here for newly graduating students from all the colleges and universities here. Thank you.
Yeah, so we're obviously as a research-focused company and eventually a development-focused company, we're looking for scientific talent, scientists to join the team, and we're tapping Penn, to be honest, and we're taking people that have worked with our program in the labs there at Penn and bringing them over to the company. That's a good source for us. And we're also looking to recruit some senior leadership outside of the Philadelphia area to come into Pennsylvania. So I think that, again, to drive a critical mass of people. And I think that as more companies locate here and bring talent in, I think that critical mass will occur.
So Laurie, thanks for the question. So for us right now, we are building fast. We're building out a management team, which, hopefully I will have completed by the midsummer, about summer timeframe. Right now, our focus is bringing industry experienced folks into the company because we have to, right? We are a clinical stage company that need to move very, very quickly. But as we now think through sort of strategic workforce planning, we've got to think 2, 3, 5 years ahead, right? And absolutely, we're going to be tapping talent in the local system at Drexel, at Penn. For example, in our company, we have to be very creative when we look at engineering as an example, right? Which isn't sort of what typical life sciences companies think about, right? Because we have to, right? Our core is going to be about how you bring clinical development thinking, scientific thinking, and blend it all into manufacturing science, right? And we have our own facilities, in northern Philadelphia, in the Norristown area. So that's going to be a big area for us as we look at the talent locally in the ecosystem. Clearly, Penn has, we've got talent now in our company that was trained at Penn, went to other companies, have now come to Tmunity, right? So it all comes together, right, in terms of the pedigree of colleagues who have been trained through the Penn system. But I think realistically, companies like ours to take and be ready to, take great talent from organizations like Penn would probably come in about 2 to 3 years as the company really takes shape and form. And frankly, our vision is, and I think I speak for all of us here, right, we're thinking about this as an investment for the region, right? It would be my vision that, in years to come, there'd be 40, 50, 60,000, 100,000 jobs here, right? That's why I committed to build a company in Philadelphia, not in Boston. So you moved. So, I mean, that's a grander scheme, right? But hopefully we can start that sort of renaissance of the life sciences sort of getting energized here again. In Philadelphia and the Pennsylvania area has a great history, right? Richness of many, many companies. And it's so diverse, right? You look at the type of companies that are here. We have diagnostic companies, medical device companies, pharma companies, big pharma, small pharma. We have a lot of talent in this region. I think what's been missing, frankly, in the past couple of years has been capital. And hopefully, going back to John's first question about is the market overheated? I don't think so, right? If the science is good, there's a lot of great talent in this city, and so in the region as a whole. So I'm really optimistic about the future here in terms of this sort of becoming a real serious biotech hub and beyond to becoming a very credible commercial hub on what is already a very successful commercial hub. I just think part of the story gets lost in Philly in the region because it's so diffuse and disparate. Boston, it's sort of all biotech-like targeted therapeutics, so people focus on that. But I don't think Philadelphia has anything less and the region has anything less than any other part of the country, to be honest. Now that I'm here and I'm looking at talent, I see it.
Just some incredible stories that you've heard here this morning. Share them. Share them with people that you know. Get the word out. You may not all be reading Nature magazine, but I can assure you, you'll be reading many publications that come in front of you about the great work of Dr. Carl June, as well as the companies that you heard here this morning. I want to thank you all for taking time from your busy schedules. Again, this is Philly Tech Week. We have a very different event tomorrow night, if you'd like to join us, where we're going to be exploring innovation in the hospitality space at Independence Beer Garden at 6 p.m., and it's going to be about 80 degrees. So T-shirts and short sleeves welcome. But we'll look forward to seeing some of you there. We also are celebrating our 25th Annual Enterprise Awards next Wednesday night at the Kimmel Center. And that is really our Academy Awards of Technology and Life Sciences Entrepreneurship in this region. And I can imagine we might see some of these entrepreneurs in a couple of years on that stage. But we'll be celebrating next Wednesday, May 9th, and would certainly hope to see some of you there. So thank you, and enjoy the rest of your day.